Hello, everybody. This is the audio recording of lecture set number 13, RT viruses, replication of reverse transcribing viruses, retrovirus. The family of retroviruses that we are talking about today are called Retroviridae. Retroviridae family of viruses have a backward nucleic acid synthesis, meaning that these viruses have a genome that consists of RNA, positive sense RNA, and the viral genome will be converted to negative DNA strand and then double-stranded DNA, positive and negative sense DNA that will be incorporated into the provirus. Retroviridae family of viruses use the enzyme reverse transcriptase, or RT, which is an RNA-dependent DNA polymerase, meaning that it uses an RNA template, in this instance viral genome, positive RNA, and makes a DNA copy out of that RNA template. So RNA-dependent DNA polymerase. The first part of the phrase refers to the requirement for template, and the second part is what would the enzyme make. In this instance, template is RNA and makes DNA. Also, there's a virus called RNase H that is DNA dependent DNA polymerase. DNA dependent DNA polymerase uses DNA template to make more DNA. We'll talk about these in a little bit more depth and detail. Slide number 3. The first subfamily is called Spumavirinae, or these are the viruses that create foamy vacuoles in cell culture. They use mammals and primates as their host, and human foamy virus was the first retrovirus found in human beings. Spumavirinae are orphan viruses, meaning that they cause asymptomatic infection, and the infection is not associated with any particular disease. The second subfamily is called Oncovirinae. As the name implies, onco, these are tumor-forming viruses, meaning that upon successful infection by the virus leads to host cell transformation. So the virus, let's say, uses a T lymphocyte as a host, and once an oncovirinae virus infects that lymphocyte, the lymphocyte becomes cancerous. We have leukemia, T-cell leukemia. So these are RNA tumor viruses, rather large repertoire of hosts, or the host range is pretty broad, includes avians, reptiles, mammals, and primates. A very famous virus in this family is called, in the family of Oncovirinae, is called human T-cell leukemia virus, or HTLV, that causes rare cancers in humans. The third subfamily is called lentivirinae, lenti, slow-growing viruses. So if it's slow, they can cause persistent chronic infections, those infections that stay for a long time and they are not acute. The lentiviruses do not cause cellular transformation. Rather, they cause chronic diseases of central nervous system, lung, and they can also affect immune cells, cause immunodeficiency. Host range includes humans, non-human primates, as well as other mammals. Prime example of viruses in this family is human immunodeficiency virus, or HIV, is an emerging pathogen, causes pandemic infection around the globe, similar to a coronavirus that we're currently facing. And the virus uses macrophages and lymphocytes as its host. Therefore, the first thing that comes to your mind immediately is that successful infection by HIV leads to immunodeficiency in infected individuals, as we see in the acquired immunodeficiency syndrome, or AIDS. Remember, AIDS is not the name of the virus. The virus is HIV, and the disease is called AIDS. Moving to slide number 6, human immunodeficiency virus. Let's look at the genome structure. We have envelope glycoproteins that are made as a result of transcription and later translation of ONV or ENV gene segment. These are glycoprotein spikes. We see it on the surface. Matrix protein comes from GAG gene segment. These will form capsid proteins that the arrangement is cosihedral and they are wedge-shaped. We have nucleoprotein, that these are group-specific antigens, come from the genome, and they are encoded by the GAG gene segment. And then we have enzymes that are encoded by the POL gene segment. These enzymes can be broadly divided into following categories. They can be proteases, they can be polymerases, such as RT and RNase H. RT is an RNA-dependent DNA polymerase, whereas RNase H is a DNA-dependent DNA polymerase. And integrase. Integrase is a nuclease. It is the enzyme that cleaves off or chops different random segments of host cell genome where the provariant can get integrated into the host cell. So integrase family of enzymes, protease families, and polymerases. Moving to slide number 7. This is a depiction of the structure of an oncovirus and a mature lentivirus. I strongly recommend you to go through the details of this slide. Slide number 8, HIV genome is a positive single-stranded RNA, yet we have 2 copies of this positive strand. And these 2 copies associate with each other by a region called dimer linkage site. 2 copies associated together by dimer linkage site. The genome is 10 kilobase, and the messenger RNA has 5' cap and a 3' poly A tail. The genome, HIV genome, contains 3 major genes, GAG, POL, and ENV, that we briefly touched on them, but we'll discuss them in a little bit further detail. In addition to these 3 major gene segments, we have complex overlapping genes in lentiviruses. These overlapping genes usually code for regulatory or accessory proteins. Some of these regulatory and accessory proteins are called VIF, TAT, REV, VPU, VPR. Moving to slide number 9. These next few slides are very, very, very important, so please pay close attention to them. Like any other genome, we have a 5' end region and a 3' end region. 5' end region of the HIV genome, which is positive RNA, has the following regions. We have an R region, U5, PB, and leader region. R, or terminal repeat, it's a region that is very important for reverse transcription by the enzyme reverse transcriptase. U5, U for unique. 5, it's a unique sequence that is at the 5' end of the genome. So we call them unique 5' end sequence. Once we make a copy, a DNA copy of this, the orientation would be switched because they are complementary. So this U5 will become 3' end of the proviral DNA. And what does it do? It signals for poly A addition to messenger RNA, or as we talked before, polyadenylation. PB is the next region, stands for primer binding site. This is a site at the 5' end of HIV genome where host cell transfer RNA will act as primer and binds to this region. And then we have leader sequence, which is a recognition sequence for packaging of genome RNA, and it's also donor site for all spliced subgenomic messenger RNAs. After the 5' end region, moving to slide number 10, we have the major central region that has 3 major genes, GAG, POL, and UNV. GAG, or group specific antigen. This gene segment codes for structural proteins. What are these structural proteins? Capsid proteins, matrix, nucleoproteins. Nucleoproteins are those proteins that bind to the genome. In this instance, the genome is RNA. So these are RNA binding proteins. The middle segment is called POL, that codes for 3 sets of proteins. Remember, PROT, POL, INT. PROT, or protease, POL, the polymerases, and INT for integrase. So protease is an enzyme that cleaves viral polyprotein. So in HIV, we make one huge protein, and with the action of proteases, that protein will be chopped off into small pieces. RT and RNase are DNA polymerases, and both are coded by the Pol gene segment, and both are required for reverse transcription. And as I mentioned before, integrase is a nuclease that cuts host cell DNA, thereby it provides the physical space for the insertion of proviral DNA into host cell DNA. And on code for envelope glycoproteins, these are proteins on the surface of the virus that a sugar moiety has been added to them, so glycoprotein. They are either on the surface or they span through the membrane. So part of it is outside and then part of it is goes through the membrane, so-called transmembrane glycoproteins. Moving to slide number 11. So far we have talked about the 5' end of the genome that contains R, U5, PB or primer binding, and leader sequence. We also talked about the central gene segments, GAG, POL, ENV. Now we are talking about the 3' end region of HIV RNA genome. The sequences in this region include PP, U3, and R. PP is a polypurine tract. You know, we have two types of nucleotide, purine and pyrimidine, T and C, pyrimidine, and A and G purines, but polypurine means that this tract, this segment has a lot of A and Gs. This is an initiation site for positive DNA synthesis of the virus. The next region is called U3, the same way that we had U5 on the 5' end. U3 on the 3' end is a unique sequence at the 3' end. This unique 3' end sequence will become 5' end of proviral DNA, the exact same way that U5 will become 3' end, U3 will become 5' end of the proviral DNA, because RNA with RT, we make a DNA. It is also a regulatory sequence for messenger RNA transcription and DNA replication. Last region in the 3' end is R, or terminal repeat, that is required and important for reverse transcription. Moving to slide number 12. HIV provirus is a double-stranded DNA, how it replicates. So virus gets into the cell. It uncoats in the cytoplasm, meaning that the capsid and the genome will be separated from each other. Viral genome is single-stranded positive RNA, but remember, there are two copies of this positive RNA that are attached together or associated to each other by at a region called dimer linkage site. So this positive RNA, with the aid of the enzyme reverse transcriptase, will be converted to negative DNA, the opposite sense. If that's positive, this one is negative, and RT uses RNA template to make a DNA. And this negative DNA strand will become double-stranded DNA that it will then be transported into host cell nucleus. And at this point, if the DNA, viral DNA, is in the host cell nucleus, the enzyme integrase, cuts host cell, virus will randomly get incorporated into host cell DNA. So how do we know that we even have viral DNA? Some people say, oh, show it to me, to prove to me that it's not just RNA. So there are several lines of evidence The first line of evidence is that the virus replication can be blocked by actinomycin D. Actinomycin D is a chemotherapeutic agent that also blocks DNA to messenger RNA step. So if we have DNA, we make messenger RNA. What do we call this step? Transcription. So blocks transcription. Also, scientists have found that those host cells that were infected with the virus, they had a DNA that was complementary to the original positive RNA genome of the virus. And lastly, when they discovered that viruses have this enzyme, reverse transcriptase, adds more credibility to the theory that viruses have RNA, but then they convert it to DNA. I should mention that the discovery of reverse transcriptase in the 70s, basically the gentleman who discovered it was awarded a Nobel Prize at a very young age. I guess it was 34 or 38 years of age. So slide number 13 is a little bit of a description of actinomycin D or dactinomycin. I would strongly recommend you guys to go through this slide. Slide number 14. Let's get to the juicy stuff. So the picture shows two different sections. The top portion of the picture shows the genome of virus, that is RNA. The bottom one is proviral DNA sequence. So what do we have in the virus? 5' end, we have RU5PB for primer binding and leader. Then we have major gene segments. Then we have polypurine tract or PP, unique region at 3, and R. So let's see what will happen. Host cell transfer RNA primer binds to the PB or primer binding site of positive RNA genome. For any of these transcriptional events, we require a primer. In this instance, at the 5' end, the primer is called tRNA primer, and the location that it binds to is called PB or primer binding. Since it is within the genome, we call it internal site. So the primer binds, and then we have the enzyme reverse transcriptase uses RNA and makes negative DNA strand. Once it makes negative DNA strand, at the same time, the remaining positive RNA strand will get degraded. So we are making from the 5' end. Then a small positive RNA primer, which is actually a little region of the genomic RNA that was not degraded, will serve as primer that binds to the polypurine tract on the 3' end and initiates positive DNA synthesis. Let's take a step back. We need to have a negative DNA strand and a positive DNA strand because proviral DNA is double-stranded. So for the negative strand synthesis, we need a primer. That primer is called host cell tRNA, binds to the PB region at the 5' end of viral RNA. Once DNA synthesis starts, we have the RT, which is the polymerase, we have the primer, we have the sequence, we make a complementary strand to this. This complementary strand that we are making will eventually become the 3' end of the viral genome, proviral genome. the remaining RNA in that 5' end region will get degraded by reverse transcriptase and RNase H. A small piece of genome RNA, the positive strand RNA, that is not degraded by RT or RNase, that small little piece of positive RNA will serve as primer, binds to the 3' end at the PP region, and starts synthesizing the positive DNA strand. So we had negative DNA strand that was started at the 5' end by tRNA primer that binds to PB. At the 3' end, we have a small piece of positive RNA that binds to PP region and makes the positive DNA strand. So this will continue. We have negative strand, positive strand. Therefore, the proviral double-stranded DNA is being made, and it has some novel ends. These novel ends are called long terminal repeat, or LTR, that include U3, R, and U5. R is common. U3 of the proviral comes from the U5 of the genome, and U5 of the proviral double-stranded DNA comes from the U3 region of the genome. Moving to slide number 15 is a description of long terminal repeats as well as retroviral integrase. I want you guys to pay specific attention to these concepts. I know that you guys have had these in your previous courses, some of which served as the prerequisite for this class, but still I want you to go through them and make sure you completely understand it. Moving to slide number 16. Okay, so if you understood the concept I told you, these two slides, 16 and 17, make perfect sense. So we're talking about reverse transcription, where we have an RNA genome, positive RNA, with the aid of reverse transcriptase enzyme, we will make a negative DNA strand. So whatever is in the 5' end of the proviral of the viral genome will become the 3' end of the proviral double-stranded DNA, and whatever is in the 3' end of the viral RNA genome will become the 5' end of the proviral DNA. Just reverse it. I also mentioned that at the 5' end of the viral genome, host cell transfer RNA is the primer that binds to the PBS, or primer binding site. Primer is there, the enzyme reverse transcriptase is there, and it makes a negative DNA strand. This negative DNA strand is a copy of the 5' end of viral genome. So it includes R region and U5. This is a minus strand. At this point, the minus DNA strand will stop synthesizing. We have something called the first jump. So the first jump is where the negative DNA strand that is being made, the R region of that will hybridize to the R region of the RNA at the 3' end. So from the 5' end, everything is jumping to 3' end of the viral genome. This is the fifth line on the diagram on the right. That small little piece of blue arrow is the 3' end of the proviral DNA. negative strand that jumps the R region of it, hybridizes to the R region on the RNA. The negative DNA strand will be extended and goes through those sequences. The sequences include U3, PP, ONV, POL, GAG, and PBS. Once it gets to PBS, the majority of the previous RNA, which is actually U3 and R region, will be degraded by RT. All is left from the 3' end of the viral RNA genome is the PP region. Está bien? So from the 5' end, we have transfer RNA as primer binds to PBS, starts synthesizing. What do we make? We make a copy of the U5 and R regions of the original RNA genome. The original RNA genome is depicted in pink, and the newly synthesized strand is in blue. Once we make it, that piece of original RNA genome, the pink region that includes R and U5 will be degraded. We don't have them anymore. Then this is the negative DNA strand, stops. Negative DNA strand includes R region and U5 region. R region of it will jump over to the other side and bind or hybridizes to the R region on the 3' end of the original viral RNA genome. So hybridization between the R region of the blue strand with the pink strand. This is called first jump. So once this hybridization happens, negative DNA strand continues its synthesis. So it makes a copy of the R region, U3 region, PP, envelope, POL, GAG, and PBS. Because PBS is where it actually started the synthesis, the third line from the top, and that's where it stops. Once it gets to PBS, everything on the original RNA genome, which is U3 and R regions, will get degraded and removed, and the PP or polypurine tract will remain. It's a region that is rich in nucleotides A and G. Moving to slide number 17, we have the second jump. The second jump is PP primer for positive DNA strand synthesis. What do we need to synthesize? On the 5' end, we need to synthesize PBS, U5, R, U3. Once we make a copy of that, that's the positive strand stops. Then the second jump happens, where the positive DNA strand will jump to the other side, to the 3' end, binds to PBS, primer binding site. So it's a switch in the template. First, it was at the 3' end, jumped to the 5' end. We extended the 5' end. Now from 5' end, it jumps back to the 3' end. And whatever is in between, which is mainly negative DNA strand, will be removed, will be degraded. And so we have negative DNA strand, positive DNA strand, both strands extend and complete their synthesis. We end up with double-stranded DNA with novel ends. These novel ends are long terminal repeats that include U5 or U3. Whatever was on the 3' end of the viral RNA genome becomes the 5' end of the double-stranded DNA genome. and whatever was on the 3' becomes the 5'. So slide number 18 is a depiction of the entire process, all the 2 steps together, binding of transfer RNA to PBS, extension of it, removal of U5 and R, stopping of negative minus strand, jumping to the other side, making a copy of the RNA all the way through PBS, U3 and R regions of the positive RNA will be degraded, PP remains there, and then we continue making the 5' end of the proviral DNA. 5'. This includes U3R and U5. Then this plus strand or positive strand stops. We call strong stop, meaning that it won't make any more, not even one additional nucleotide. Then it jumps over to the 3', and we call it second jump or second template switch. And the intervening sequence, which is mainly negative DNA strand, will be degraded. Then we end up with double-stranded, positive and negative, 5' to 3', 3' to 5' of proviral DNA with unique sequences called LTR. I have included 2 useful links for you, and I would definitely encourage you to take a few minutes and go over these YouTube video clips. Slide number 19 is a detailed description of the replication of retroviruses. Please take a few minutes to read it. It is super simple, but it may sound a little difficult to some individuals. Therefore, I strongly recommend you guys, once you're reading this and going through it, make sure to have a piece of scratch paper in front of you and start drawing it so it makes sense. Slide number 20, how does the RNA retroviral genome gets expressed when we only have one promoter to make GAG-ONV proteins and Pol proteins. How this is done, please take a few minutes to understand this, and then you will refresh your memory on the AUG translation, start codon, a little bit about the proteolytic cleavage by the use of enzymes, proteases. Moving on to slide number 21, HIV provirus integration into host cell DNA. At this stage, if we have HIV provirus is DNA. So at the ends of proviral DNA, we have long terminal repeats that is required for this integration. We also need the viral integrase, the enzyme that is a nuclease. Since it's inside of the cell, we call it endonuclease, that nicks or creates a little cut on the host cell DNA at random sites. It's not a very specific or site-specific cleavage. It's a random site, and viral DNA will randomly get inserted into host cell DNA, and then the enzyme ligase will patch it up and like glue sets it in place. This integration is absolutely required for the infection caused by retroviruses, and whatever is outside, whether it's viral RNA, viral DNA, complexes of these will be degraded by host cell enzymes. So there's no leftover. Moving to slide number 22, HIV provirus mRNA transcription. The transcription of mRNA requires host cell RNA polymerase II, and we make a genome length messenger RNA. This one single genome length, so the entire genome, this one messenger RNA translates for 3 proteins, GAG, POL, and ENV. Sorry, let me take a step back. It encodes or translates for GAG genes or GAG and POL genes and then proteins. And the way these proteins are being made from one messenger RNA is by translational frame shift. And then multiple splicings occur to create subgenomic messenger RNAs. Each of these subgenomic messenger RNAs code for one of those proteins that we talked earlier on. And these are required for assembly and preparation of the progeny for provirus. For the assembly and maturation of the genome of the progeny virus. So slide number 23 shows HIV spliced messenger RNAs. As you can see, we have multiple messenger RNAs. They all have 5' cap and 3' poly A tail. The 5' and 3' is identical. The rest of it is different. So it translates for envelope proteins and also for a series of regulatory and accessory proteins. ENV protein will become the envelope of the virus, and these regulatory and accessory proteins participate in the switch for subgenomic messenger RNAs. They will also favor or assist chronic infection of HIV virus. They downregulate NEF protein, activate another protein, and cause the activation of an infectivity protein, or VIF. It's a protein that is required for the pathogenicity of the virus. Moving to slide number 24, HIV assembly and release. The virus releases progeny RNA in cytoplasm. Capsid proteins will be assembled. They insert genome RNA, and this genome RNA will migrate to virus matrix proteins. Then envelope proteins are being made, they are glycosylated, so those envelope glycoproteins will be inserted into a specific region of the plasma membrane. And since it's modified by the virus, we call it virus-modified host cell plasma membrane. And once the virus is exiting from the host cell, it picks up a piece of the host cell plasma membrane as its envelope, and the release is by budding. Slide number 25 is a summary and depiction of everything that we discussed so far, so I would strongly recommend you to take a quick look at it. Slide number 26, HIV pathogenesis. Virus uses macrophages, which are phagocytic cells of the immune system. These are the cells that phagocytose foreign objects and present it to T lymphocytes or B lymphocytes. So virus infects those as well as CD4 positive helper T cells. CD4 positive helper T cells regulate both humoral and cell-mediated immune responses. So they're key players because they secrete 2 types of cytokines, type 1 cytokine, type 2 cytokine, with a myriad of effects. The next stage in HIV pathogenesis is persistent chronic infection. So an infection that doesn't go away and lasts for long periods of time, and it's in the lymphoid tissues of the host. At this stage, we see clinical symptoms of PGL, or persistent generalized lymphadenopathy. Lymphadenopathy is the swelling or enlargement of the lymph nodes. Even though virus is in the lymph nodes, but virus is held in low copy numbers by host defense mechanisms. Unfortunately, over time, virus resumes its replication at a very high level. It destroys its hosts, which are T cells and macrophages. Therefore, host immune response is compromised. This is the time that we see clinical AIDS disease, and due to impairment of the host immune response, opportunistic infections such as a myriad of viruses, pneumocystis carinii, AIDS-related lymphoma, etc., will ensue and result in the death of the patients. The way we follow the course of HIV virus infection is by counting the number of CD4 positive T cells, which are not only a critical player in cell-mediated and humoral immune response, but also these are the hosts of the virus. We do PCR to measure HIV mRNA load in the patient's serum, and will look at the clinical signs and symptoms of the disease in patients. Slide number 27 depicts what I just mentioned on the y-axis. On the left side of the picture, we have CD4 count. On the right, we have RNA copy number per milliliter of plasma. As you can see at the beginning of the disease, where we have an acute infection, CD4s are high and virus is in check, viral load. But then as disease, for some time, virus load is low because the immune system is still functional. Then virus keeps infecting more cells. We initially see PGL, persistent generalized lymphadenopathy. Slowly and gradually, the number of CD4 positive T cells will decline. That's where we have subclinical immune dysfunction. And once the CD4 positive cells are very low, most of these cells are destroyed by HIV infection inside of them. And HIV replication is so fast, viral load starts going up. That's where we see clinical AIDS and opportunistic infections such as Candida albicans, EBV or Epstein-Barr virus infections, CMV, cytomegalovirus, tuberculosis, pneumocystis carinii, and other infections that will eventually lead to the death of the patient. Slide number 28, origin of HIV. The oldest documented case goes back to 1959. And they thought that in West or Central Africa, simian virus infects non-human primates, and it's very similar to HIV infections. Therefore, they came up with this theory that there is a cross species, meaning that infection of SIV by infected blood and then its transmission to other humans. Also, Bushmen hunt animals such as monkeys and apes for food. They slaughter them. Once they kill them, they come in contact with the raw meat, blood, and bodily fluids, and that's a source of cross-contamination. Slide number 29, emerging viruses. HIV is an emerging virus. In 20th century, there were changes in Central Africa due to social, economic, and behavioral matters. Rural migration to the cities, increase of trade and commerce between distant regions of the globe, and that would result in less farming, and people would rely more on bushmeat for food. And Kinshasa Highway was dubbed as the AIDS Highway because there was a coast-to-coast traffic. Male truck drivers were working there continuously, and there was a growth of sex trade leading to HIV dissemination. And also another reason for emerging HIV is the breakup of traditional family units. We have a lot of single female parents, male parents working in city, sex outside of marriage, etc. Slide number 30 is just basically showing, giving an estimate of number of people per million infected with HIV in different regions of the world. This chart was prepared by WHO, or World Health Organization. In 2013, there was a pandemic of HIV or AIDS, mainly heterosexual transmission by blood, bodily fluids, sexually transmitted diseases, and drug abuse. And since the start of epidemic, 78 million individuals have been infected, and almost half of these, 39 million deaths. In 2013, there were 35 million individuals living with HIV infection or AIDS disease or the syndrome. 1.5 million of these died of AIDS-related illnesses, 2.5 million new infections. Many babies are infected perinatally, moms are infected, so on and so forth. Moving to slide number 32. The next few slides talks a little bit about the oncogenicity or cancer-causing properties of retroviruses. These are RNA tumor viruses. They have their own oncogenes. Oncogene is a gene that encodes proteins, that these proteins can cause cancer. So cancer-causing genes. We have V-oncs, or viral version of an oncogene, and C-onc, which are cellular versions of the same gene. And most likely, V-oncs are subverted from cellular genes, or C-oncs. So oncoviruses, the viruses that cause cancer, use three major mechanisms to cause cancer or cellular transformation. These include production of oncogene transforming proteins, altering host cell regulatory networks, and stimulating host cell growth. We'll talk about these three scenarios, but know that these oncoviruses provide useful models to study host cell regulatory systems and transformation events. And but also remember that most human cancers are due to chemical carcinogens, and viruses serve as co-carcinogens. Moving to slide number 34, the first way by which oncoviruses cause cancer is by the production of oncogene transforming proteins. This causes rapid transformation. Example would be Ross sarcoma virus in chickens. And we have a viral oncogene called SARC. This viral oncogene protein is a tyrosine kinase, meaning that it adds phosphate groups to tyrosine residues, resulting in constitutive activation of key regulatory proteins in signal transduction pathways such as metabolism, and lead to rapid transformation of the host cells or cancer. Slide 35. The second method is by altering host cell growth regulation. This is a slow transforming process. The second difference is that the virus in this instance does not have oncogene, whereas viral genomes such as murine leukemia virus, MLV, integrates into host cell DNA. Once it integrates into host cell, it turns on some cellular oncogenes such as growth regulatory or growth control genes, and these will result in continued cell activation, and over a long period of time when the cell cycle is disturbed and is continuously active, results in cellular transformation. The third mechanism by which oncoviruses cause cancer is by stimulating host cell growth factors. This is again a slow transforming process, and virus does not have oncogene. Example would be HTLV, which we just talked about, human T-cell leukemia virus, that infects T lymphocytes. Once a T cell is being infected, they continuously release cytokines, same concept that we talked about with interferon and interferon receptor and antiviral effector molecules. So continuous release of cytokines stimulates the growth of the neighboring T cells. Over a long period of time, we have continuous T cell activation that leads to cancer of T cells. The last concept that I want to discuss is the retrovirus-like genetic elements. There are some genetic elements that are similar to retroviruses. Between the years of 1920 to 1980s, Barbara McClintock was doing research on the cytogenetics of maize, and in the 1940s, she proposed a concept called transposition, which are these gene segments that can move around, transport. They can jump from one area to the other one. They're called movable genes. Once these genes are moved from one location to the other one, they turn maize traits on or off. So they basically modify the genetic profile and the phenotype. At the time, this theory received widespread skepticism. Everybody thought that she's out of her mind. But later on, when researchers were working on retroviruses, found a similar concept. They saw that there are some genes that if you remove them and put them in retroviral genetic elements, they can allow or they pave the way for genetic diversity. And these movable elements are not only in retroviruses. We have them in bacteria. We call them bacterial transposons. That these transposon or movable genes cause resistance to antibiotics. We have retrotransposons in yeast and Drosophila, and we also have retroposon in humans. So after being bombarded with all this skepticism, in 1983, Barbara McClintock received Nobel Prize for her research, which just basically shows her dedication, perseverance, and more importantly, believing in herself. Retrovirus summary. The members of retrovirus are spumovirus, orphan virus, they do not cause any disease. Oncoviruses, tumor-causing, and lentivirus cause persistent chronic infections. Retroviruses have three major genes: GAG, code for capsid proteins, POL for enzymes, for envelope proteins, and the envelope proteins will later on become glycosylated glycoproteins. So proviral double-stranded DNA is being made from the virus messenger RNA genome or positive RNA genome by the use of enzyme reverse transcriptase. Remember, we talked about the 2 jumps. We start at the 5' end of the RNA, then we go to the 3' end of it, then connect the 2, get rid of the extra RNA or extra negative DNA, and we have double-stranded DNA with novel ends, long terminal repeats. Then cell-integrated proviruses use RNA polymerase II for messenger RNA transcription. Once the virus gets in, it needs host cell RNA polymerase II for the transcription of mRNA. Once the mRNA is being made, then it undergoes proteolytic cleavage or processing to make all these enzymes, and we talked about the creation of subgenomic messenger RNAs. There are 3 ways by which RNA tumor viruses can transform or cause cancer in host cells. These are the use of viral oncogenes, turning on host cell oncogenes or c-onc, or stimulate cell growth pathways such as infection of T cells and continuous secretion of cytokines. There are genetic elements such as those found in retroviruses that can move from one area to the other one and turn on or off some traits. These are called retrotransposons or transposons, and we can see that in various cell types: bacteria, yeast, Drosophila, and humans. Slide number 40 shows the required reading for this lecture. All come from chapter 19, retroviruses, converting RNA to DNA. This is the end of audio recording for lecture set number 13, RT virus. Thank you so much for your attention.